Hello, everyone, and welcome to the TrialSite News Podcast called Farrago. I am Erin, your host, and thank you so much for joining in. We are here with a special guest, Dr. Denise Faustman, and thank you, Dr. Faustman, for joining us today.
Well, thank you for inviting me.
Absolutely. And for folks at home, we are going to be chatting about the BCG vaccine, COVID-19, particularly in type 1 diabetics. But first, Dr. Faustman, do you mind taking a few minutes to introduce yourself and tell our viewers the type of work you do?
Okay, sure. So I'm an MD PhD, which means I went to school forever before I had a real job. And so our favorite thing to do is the basic science of a project. And then because we look at data very critically, know whether we believe it's going to go to human clinical trials, and then actually do the clinical trials. So the study we're going to talk about today is actually a phase 3 randomized, double-blinded clinical trial that was conducted here at the Mass General Hospital.
Checks all the boxes, right? Double-blinded, placebo-controlled, the whole works.
Yeah, randomized.
Randomized.
Yeah, for those people who don't know what those words means, it means half the people got the vaccine and half the people didn't get the vaccine. And everybody involved is blinded until the trial's done. So there can't be a bias along the way. Yeah.
All right. So we're going to talk about the BCG vaccine. Do you mind just telling our viewers a little bit about the vaccine, just a general summary and its history? Great.
Yeah. So first, I always like to give credit to people in the scientific world who contributed greatly to this field of the BCG vaccine. So this vaccine is 125 years old. So talk about an old vaccine. That's what we're talking about today. Known by the WHO and other organizations as the safest vaccine ever created on the planet. And for the last 125 years, it's been given to literally billions of people. So in the US, people will say, what are you talking about? BCG vaccine? Is this the Boston Consulting Group? And you go, no. But if you're from India, China, South America, Africa, you're going to go, of course, the BCG vaccine. So it was originally developed for tuberculosis. And it's so high tech, just so you know how this was created. It's literally the scrapings from cow udders. Okay, this is the same vaccine. So this is tuberculosis of cows. And of course, they discovered that the milkmaids that were immunized from this didn't get TB. So why are we studying this today, 125 years later? So the reason we're studying this, and other people are studying it, and these studies, really, I've got to give my European colleagues a big applause, because they've been picking up the last 10 to 15 years work called off-target effects of BCG as it relates to infectious diseases. So with some randomized trials that were first started out of Denmark and conducted in Africa, they realized that infants that got this had lower morbidity and mortality, like a really big signals. And infants at birth who did not get this vaccine had higher mortality, very statistically significant work. So people around the world have been trying to see if this vaccine could be used like as a platform vaccine, like in the US. And the US is a particularly good clinical trial subject group to work on, because in the US, we've never had the BCG vaccine. Okay, it's never been licensed here. It's not available at birth. You've never gotten it. So this means when we recruit people that are born in the US, raised in the US, they don't have prior exposures to this vaccine. And to combine that with our study, we want to look at infectious disease vulnerable population, and those are people with type 1 diabetes. Okay.
And just to go back to the word off target, just so folks know what that means.
Yeah. So if you said on target, that would mean we're giving people in the US BCG and saying, do you get tuberculosis? So off target means secondary effects of vaccines that might be important and beneficial. And some people call them side effects, but these are like highly desirable side effects that are maybe even more important than its tuberculosis protecting fashions. Yeah.
And we're going to dive into how you did your study. But from what I read, is it true that the off-target effects against infectious diseases, there's something about it takes 2 years?
Yeah. So this is
Okay.
a very important clinical trial design point. So it's not just doing a clinical trial, it's understanding how many doses you need to give in a clinical trial, how long it takes to kick in. And probably I shouldn't be calling it a BCG vaccine. I should be calling it a BCG immunotherapy. Because when you hear the word vaccine, you think, oh, somebody's going to give me something, and 2 weeks later, I'm going to have protection. And that's not the case, because this vaccine, which is a live bacterial product, actually resets the immune system. So that takes a couple years to kick in, especially in not previously vaccinated adults. But it may also, on the other side, provide lifelong protection from infectious diseases. It's known that if you get a BCG at birth, it takes a couple of years to kick in to protect those kids. And those newborns, like in Africa, don't get exposed to TB till they're 10, 12, 15, 30 years old. And the vaccine's still working. Okay, so it's the word vaccine kind of biases you to think, well, it's got to a while to re-educate the immune system.
Hey, we lost you there for a second. But
okay, yeah, I didn't know who was losing who.
Right. Well, isn't that always the case? We're still talking about the vaccine here.
Not another interview.
Right. And the off-target effects, when you say protective effects against infectious diseases, is that an umbrella term for just a lot of other infectious diseases, or?
Yeah. So what
Okay.
people globally have seen really over the last 10, 15 years is they do trials for malaria, or they do trials for yellow fever, rare diseases. And sure enough, the results were a lot better if the people had gotten the BCG vaccine. And then even the Gates Foundation was trying to develop a new BCG vaccine for none other than tuberculosis. And the old-fashioned BCG vaccine, instead of the new recombinant, showed the people who got the old-fashioned vaccine weren't having respiratory tract infections. So time and time again, many, many people globally have been seeing this platform infectious disease protection.
All right. So this study was over a 19-month period conducted here in the US, like you already mentioned, late in the pandemic.
Yeah. So remember, late in the pandemic. So why would I use that word late in the pandemic versus early in the pandemic? So probably everybody remembers when COVID was here, as it started to evolve, it killed people, but it wasn't very infectious. But then as COVID evolved in the US and also globally, it killed less people, but it was highly, highly infectious. So you could create a vaccine early in the pandemic and it wouldn't work late in the pandemic, or vice versa. So this was really important to follow these type 1 diabetics who had in the previous years gotten 5, 6 vaccines of BCG to see what was the impact of that on COVID and what was that impact overall in infectious diseases. Okay. So a very special population from the US, vulnerable to infectious diseases, okay, particularly susceptible to COVID, and lifelong have incredible morbidity and mortality from infection. So it was better than using healthcare workers for a COVID vaccine because their incidence of infectious diseases is so high.
And that's just because folks who have type 1.
Yeah, it's type 1. It's specific to type 1, not type 2.
Okay. And that makes more prone to getting
all infectious diseases. Yeah, like gangrene of feet and amputations, like bladder infections, like colds, flu, on and on.
All right. So what sort of results did you see?
Yeah. So what's the answer? So here comes the answer. So the primary outcome was for COVID infections and for infectious diseases over this time period were people with diabetes who got multi-dose BCG less likely to get COVID and less likely to get infectious diseases. And the data overwhelmingly shows in this US population, their incidence of COVID was down, and dramatically the incidence over that time period of all infectious diseases was also down compared to placebos who were type 1 diabetics in the general population.
And this was a similar result to your previous trial.
That's right.
A phase
so
2
varied
trial.
a little bit. The previous trial was early in the pandemic. Okay, lethal COVID, no vaccines, no commercial vaccines available. Right? I mean, just so we were able to show in that trial that the efficacy of BCG was equal to the Pfizer and the Moderna trial. Okay, equal. So now we got to this trial, and this trial was a little more complicated, right? Because the viruses had changed, more infectious, and also a little more complicated because the mRNA vaccines were out there. So what we also had to study was, one, the fundamental question, do the commercial mRNA vaccines work in type 1 diabetics? Do the commercial mRNA vaccines help BCG, or do the commercial BCG vaccines hinder BCG? So 3 additional things were reported on this study. One is, and we're not the first group to show this, again, I like to acknowledge my colleagues, the mRNA vaccines had no efficacy in the type 1 diabetic population, okay?
None at all.
Not at all. Not at all. Okay. And it's believed that it's because in type 1 diabetes, it gets a little geeky, but they don't have good T cell responses. They don't have good CD8 responses. And to get a good vaccine response, you need the B cells making immunoglobulin and you need the T cells. So they don't have the T cell part. So we confirmed some other work that was done at Children's Hospital here in Boston that type 1 diabetics were unresponsive. And then everybody in the vaccine community wanted to know the next loaded question. Did the COVID vaccines harm BCG or did the COVID vaccines help BCG? And the answer is none of the above. They didn't help BCG, but they didn't harm BCG. There's some data globally that when you get a live vaccine like BCG, you don't want to get a bunch of dead vaccines afterwards. But our data in the US in this patient population did not see that undesirable side effect. We just saw no effect of mRNA vaccines on helping or hurting the BCG effect.
But with BCG vaccine, those who have type 1 diabetes were less likely to get sick with COVID-19.
That's right. That's right. Symptomatic and confirmed by PCR.
Okay, so the less severe sickness as well.
That's right. That's right. Our study was not powered to look at morbidity. Okay. In other words, did you die or not? There are studies that are globally doing that directed by Christine Ben in Denmark, looking at, yeah.
She's great.
Yeah, and she published, there were some groups that should be acknowledged that did COVID studies that in Europe mostly and in Africa that said, oh, we don't see anything. But there's some important distinctions to make. One is if you go into a population like in Africa that already got BCG at birth and BCG lasts for 70 years, how can you prove an effect of giving BCG in a COVID pandemic? Okay, you're not going to see it because your placebo population has it. And the other thing, because it was pandemic, everybody was right around trying to conduct these trials, forgetting that in adults, it takes 2 to 3 years for this vaccine to kick in and then confer lifelong protection. So there's some pretty good differences in trial design that are a reminder that maybe we shouldn't be calling it a vaccine, but be calling it an immunotherapy. public health is way beyond what I can do. Okay, I know it's your background, but it's way beyond what Denise Faustman can do. But it does bring up the point that if people are healthier with this lifelong protection, we should be thinking about this in the US for more prevention therapy for the next pandemic. But that's out of my pay grade, okay? So we're moving forward with our studies to protect type 1 diabetics.
All right. But I will take a note of that and present it to my public health colleagues.
Okay, yeah. Like, why aren't we doing this? It's simple, it's cheap, it's safe.
It's cheap too, right? That's an important point because the mRNA vaccines were not
affordable healthcare in the U.S.
Right.
But don't
right,
go there.
Right, yeah, right, yeah. I mean, that's well, goodness gracious, that's a whole other podcast.
Exactly.
So, you mentioned this at the beginning that the BCG vaccine reprograms the immune system. Can you talk a little bit about that? How it does potentially have these types of effects?
Yeah, so now I think you're from California, because everybody from California wants to know, like, why is this vaccine doing this? Like, why is this organism?
I'm from small town Pennsylvania.
Okay, well, you're not far enough west, okay? You gotta go further west.
Okay.
And this is the way to think about it. And probably your audience knows that we study the effects of this vaccine on autoimmunity too, okay? And other people in Europe study the effects of this vaccine in multiple sclerosis. So there's a lot of people looking at the benefits of reintroducing this ancient organism. So about 1985, or a little bit before that, there was a group in England that started this hypothesis called the hygiene hypothesis. And it's exactly the hygiene hypothesis. And what they noticed is allergies, infectious disease, and autoimmunity were more common in wealthy communities with small households, not 16 kids, who did not live and were not raised in a rural environment. So in contrast, if you were raised on a farm and you were exposed to animals all your life and you were digging in the dirt effectively, you didn't get autoimmunity, you didn't get allergies, and you didn't have food, eczema, etc. So there's maybe a little truth to that observation, because it's now known BCG, which is tuberculosis a la cow, right? The safe version, was known to infect Neanderthals. So when all these guys go out and find a little DNA from a Neanderthal and they start sequencing, they're hoping to get any DNA, and inevitably they're getting tuberculosis DNA. So back in the most ancient times, 100,000 years ago, people evolved with BCG in them. Okay, now you don't want BCG in you today.
Right.
But some of these other organisms really worked synergistically with you to reset your immune system, and vice versa for your immune system to reset the bug. And as we've gotten cleaner and cleaner in our environment, that synergy doesn't exist. So effectively, why I think the world is seeing these beneficial health effects of reintroducing this microorganism, ancient microorganism, is that's the natural thing. And what's not natural is not to have this in your system. So we're not a big fan of hoop transfer studies and things like that. But we think this ancient organism so evolved so closely with making people healthier is just one way to create the non-hygiene that we need for a better immune system. So that's the California Talk 101. Okay.
Well, I think everybody appreciated that, even if they're not in California. Good information to sit with, and folks can dive in deeper.
Think about it. Yeah.
Think about it. Exactly.
For the people come in and go, like, I can't get it. So then it's the conversation, get a dirty dog, get a big dirty dog, and put a garden in your backyard, don't just live in a sterile apartment.
Right. That's, I grew up with lots of animals My father's a veterinarian, so
There you go.
yeah,
There you
was
go.
exposed to a lot.
You got natural protection.
I do. Yeah. Yeah. My first dog had hemophilia, and sometimes he'd have to transfuse him on the dining room table. And
Oh my
we just,
God.
yeah, it lots of different animal rescue.
A
So
lot of exposures.
skunks, squirrels, you name it. So I have to ask, so you mentioned you did the study in those who had type 1 diabetes. Now, what are your thoughts on how well this would work in those who don't have any known underlying health issues?
Yeah, I think it would work very well. Now, when you go to do a trial like in healthcare workers or the general public, you would have to make that trial really big, right? Because over a month, who gets sick is going to be a lower denominator. So those trials can certainly be done, but they've got to be huge trials and longer time periods.
All right. So final question here for you, but feel free to chime in with whatever else you want at the end of this. The biggest public health take-home points from your research, what would you say they are?
Well, they're mostly directed to type 1s. I mean, you can't always generalize to another population.
Right.
But for type 1s, there's going to be a lot of health benefits. You know, we're going to be reporting about a year from now, a 5-year-long trial in diabetics as it relates to diabetes outcomes. That kind of data, when you do randomized clinical trials, is going to show what about dose, what about time from vaccine to efficacy. Certainly, I know in the US there's a lot of resistance to vaccines, so I like to call this immunotherapy. And then I'd like to always say the supplies are always short, so this will never be made mandatory. So then everybody lines up at the door once you say that. So, and it's known to be safe for 100 years. So it's, to me, kind of a no-brainer that you should be using things in humans that have that kind of safety record. That have such exponential health benefits.
And this has a very long-lasting immunity.
Yeah. For other outcomes, we followed people 15 years. And these trials, remember, they were 18 months and 34 months. In the COVID world, those are long trials. I mean, the original Pfizer Moderna trials were 3 months long. So the advantage of these trials in the COVID world is we spanned every viral variant of COVID in the US. I mean, there's some new ones coming now, as I'm sure you're reporting on, but no mRNA vaccine or other technology vaccines been able to cover all those variants. So that's very encouraging to try to make a healthier population.
Right, 100%. I mean, and that's sort of been the problem, the main issue with the vaccine.
How do you chase the variant?
Right, cat and mouse,
To England, are you going to get a different variant? And the vaccine lasts 3 months. So this is a very different approach to protecting from infection.
Right, and a lot of people are very interested in safety, and this has and this has a very long, sounds like, safety record. Durability and safety. Yeah. All right. What are you working on next? You mentioned a 5-year study.
Yeah, the next reporting out, probably about a year from now, will be a 5-year study looking at the off-target effects of 6 doses of BCG on blood sugar regulation. That's another known effect of tuberculosis. When you get tuberculosis in your lungs, it resets in that ugly thing in your chest x-ray called a granuloma, the lymphoid system, to aerobic glycolysis, which means sugar utilization. So now with giving BCG systemically, we're looking to see how it resets lymphocytes to eat sugar. Okay. So more to come.
More to come. All right. And we would love to have you back on the podcast. It's always a pleasure chatting with you.
Yeah. Nice to meet you too.
Yeah, absolutely. And to our viewers, I hope you subscribe to the TrialSite News podcast and check out the articles on trialsitenews.com. There's opinion articles and there's journalistic articles, and you can comment and make your views known. And thanks for tuning in today. Dr. Faustman, thanks so much.
Oh, thank you for asking.
Absolutely. It's very interesting research.